All right, we're going to give it just a moment here
Okay.
before we get started. All right, looks like we have some attendees rolling in. Welcome. All right, so I think we're going to go ahead and get started here. So again, welcome everyone to the 2023 PI conference. My name is Julie Magnuson, and I will be your moderator for today's session, Genome Editing for Wiskott-Aldrich Syndrome. I'm excited to welcome Dr. Porteous, who will be presenting this topic. There will be time for questions at the end of the presentation, so please submit any questions you have to the presenter in the Q&A section of the platform. Dr. Matthew Porteous is a professor of pediatrics specializing in stem cell transplantation at Stanford Medicine Children's Health. Thank you so much for joining us, Dr. Porteous. The floor is all yours.
Thank you, Julie, and thanks everyone for joining us today. I will just pull up everything. Okay, it's a real pleasure to be able to speak to you about where we are in terms of using genome editing for primary immunodeficiencies and specifically for Wiskott-Aldrich syndrome. I look forward to your questions following my presentation. And I hope that I've targeted this presentation to bits of it, at least to everyone in the audience. Genome editing is a very hot field, and so I have the opportunity to engage with several biotechs. However, everything I talk about today is from my own academic lab or from published in the academic literature. As you're all probably aware, monogenic diseases, diseases that are caused by inherited changes or mutations in single genes permeate all of medicine. It's estimated that there are maybe at least 6,000 such diseases, maybe even more. And while almost actually all of these serious genetic diseases are classified as rare in the United States because they affect less than 200,000 people, in some, they affect millions of people in the US and millions of people worldwide. And again, this is an audience that probably does not need convincing that it's not the impact of such diseases goes beyond the single patient, because there's an echo of the disease that affects the family members and communities around them. They also disproportionately affect children. And so in terms of quality of life years, they have a disproportionate impact. And finally, a few years ago, a study was published that showed that in the United States alone, the impact of rare diseases, of which most are monogenic diseases, is on the order of $1 trillion per year in both direct and indirect costs to the United States economy. Now, genome editing is an evocative term because it suggests that we can correct these mutations in the genome like we can correct a document on our computer. And I'll highlight that this is different than lentiviral gene therapy, in which a virus is used to introduce a new copy of the gene, but that new copy of the gene lands in the genome in an uncontrolled fashion. So we don't control where it goes. Whereas in genome editing, we can correct a typographical error, such as correcting a disease-causing mutation. We can add a full sentence, and I analogize a full sentence as if it's a full gene. We can add a new sentence and functionally delete the old one. So for example, the genome the person may be born with might be read as the SCID gene makes a non-functional protein. But after genome editing, we can edit this sentence to say the SCID gene makes a functional protein. It leaves the non-functional words intact, but they're functionally crossed out because they're no longer read by the cell. We can replace one sentence with another sentence, and we can add clauses to sentences. But what I'm going to talk about today, because it's most relevant for Wiskott-Aldrich syndrome, is this idea that we can convert the SCID gene makes a non-functional protein to the SCID gene makes a functional protein, or the Wiskott-Aldrich syndrome gene makes a non-functional protein to the genome now saying the Wiskott-Aldrich syndrome gene now makes a functional protein. The way we do this is there are lots of different ways of doing genome editing. At the end, I will discuss why some of the others are not going to be applicable for Wiskott-Aldrich syndrome. But this form of genome editing using CRISPR-Cas9 is the way that we think we can address diseases like Wiskott-Aldrich syndrome and other primary immunodeficiency diseases. And the way it works is that we use a Cas9 protein, which is derived from the CRISPR system. This is a molecular scissors, meaning it can cut DNA, the two strands of DNA, to create a double-stranded break. The molecular scissors does not know where to cut the DNA until it is complex to what we call a guide RNA. And the guide RNA is called the guide RNA because in designing it correctly, it can bring the Cas9 molecular scissors to the exact site of interest in the genome. Here I've labeled it as the target site of interest. The double-stranded break is created, and that activates the cell to repair the break because damaged DNA will cause the cell to die. And there are a number of different ways the cell can repair the damaged DNA. And the way that I will focus on is on the right-hand side of the slide, which is by homologous recombination or homology-directed repair. And by providing a piece of extra DNA that looks like the damaged DNA to the cell, but has sequence changes that we want to introduce into the genome. So in this site, we want to convert the target site of interest to the target site of great interest. The cellular recombination machinery will use that undamaged donor template via copy and paste mechanism to convert a target site of interest to a target site of great interest. And this is the system that we use. Now, the reason we want to target our genes exactly where we want is that for many of the primary immunodeficiency diseases, it's not just giving the gene back to the genome, a good copy of the gene back to the genome, but we need that good copy expressed, utilized by the cells at just the right time and in just the right amount. And there are a number of different diseases associated with primary immunodeficiencies or PIDs that the regulated expression is absolutely essential. And so these include the gene that's involved in severe combined X-linked SCID, IL-2RG, and these include the gene involved in Wiskott-Aldrich syndrome. And I'll show you data that how when you target the gene, the good copy of the gene exactly where you want, you actually get you get a better effect. The system we use is, for those technical aficionados in the audience, is we engineer blood stem cells outside the body. And we do this by introducing our CRISPR-Cas9 as a protein guide RNA complex. And then we deliver that correction DNA on a non-integrating viral particle. Now, I want to be clear here that the virus we're using doesn't integrate in the genome. It is designed to introduce the DNA into the cell, but as the cell divides, the viral DNA disappears. And this is a highly efficient system and allows us to engineer the stem cells of the blood with frequencies, as you'll see, of 40 to 50%. So the first example I want to give was work done in my lab by Dr. Mara Pavel-Danu, and it was focused on X-linked severe combined immunodeficiency, sometimes called bubble boy disease, because of the relatively famous example in Houston. This is a disease in which mutations that cause the severe combined immunodeficiency, in which boys are missing both T cells and B cells, are scattered throughout the genome. And so the concept of designing a system to fix every individual mutation is possible technically now, but is not possible from a regulatory standpoint. And I'll talk about that a bit more later. So the system we designed was, let me just step back, is we want to correct this mutation, not in the lymphocytes or the precursor to lymphocytes like T and B cells. We want to ultimately correct the defect in the hematopoietic stem cell. The hematopoietic stem cell is the cell that can give rise to lymphocytes and other blood cell types for the entirety, for the duration of one person's life. So if one can give corrected stem cells to a patient, they will then have corrected lymphocytes and platelets for the rest of their life as well. So the system that we developed was a hybrid gene therapy genome editing system. I call it a hybrid system because it's gene therapy in the sense that we're taking a good copy of the gene called a cDNA, but it's genome editing because instead of inserting it into the genome in a random fashion, we insert it exactly back into the gene itself, so that now the good copy of the gene is going to be used instead of the bad copy of the gene, but all the natural factors that regulate when and where and how much the gene should be expressed are intact. So what we see for SCID-X1 is that we'll get low levels of expression in the cells that have low levels of the gene and much higher levels of expression in cells that need higher levels of the gene. And doing this, we show that we can reconstitute signaling through the receptor by looking at downstream targets. And this is one of the important points I want to make is that, and it's on the right-hand side. So in collaboration with Succeed Raven and Harry Malik at the NIH, they gave us cells from a patient with SCID-X1. And we ran them through the genome editing process I described, and then measured whether the corrected cells would develop B cells and T cells, and T cells. And what we found, and NK cells. And what we found is that in blue here, when we put the human corrected cells into a mouse, you see that we got T cells labeled by a marker called CD3, and we got a small number of NK cells. But when the cells were not corrected, shown in white, that there was very little development, if any development, of T and NK cells. On the right-hand side, Succeed and Harry and their team did a comparison where they compared how many NK cells were made if you did gene correction using AAV, labeled here as AAV, versus correction using a lentiviral vector, where the gene gets inserted randomly, and that's labeled here as LYC. And they measured the amount of NK cells that were developed after those two types of genetic engineering. And you can see in healthy donor cells, so cells from patients who don't have the disease, you get a certain number of NK cells in this assay. From the patient, there were no NK cells made consistent with the disease, but with the lentivirus labeled LYC, there was almost no NK cells made either. It was only when you inserted the gene back into itself that you were able to generate NK cells, going to the fact that by inserting the gene exactly where you want, you not only avoid the risks of genes being inserted in the wrong place in the genome, but you get enhanced efficacy as well. We showed that when we modified human hematopoietic stem and progenitor cells, that we showed through studies in mice that we've modified actually the long-term hematopoietic stem cell that would end up giving rise to healthy lymphocytes through the duration of someone's life. So I'm going to get to where we are with the SCID-X1 program. So a couple of years after we published our paper on SCID-X1, Alicia Cavazza, along with Adrienne Thrasher and her group, applied a similar strategy to Wiskott-Aldrich syndrome. And again, what they did is they used the CRISPR AAV system I described to insert different forms of the Wiskott-Aldrich syndrome gene into itself. And they compared how effective that was compared to using a lentiviral gene therapy vector shown as number 3 on the left as WW1.6. And what they showed, interestingly, on the right-hand side is that you could get as more Wiskott-Aldrich syndrome protein cells, so more cells expressing the protein using the gene editing system shown as WPA, than with the WW1.6 system. You can see that the WPA, the number of cells, is higher. So it shows us it's just as effective as inserting genes as it is as a lentiviral gene therapy vector. So then they wanted to look at, well, after the cells were corrected, did the macrophages and platelets, two of the cell types that we can derive in a test tube, how were they fixed between the two systems? And what they found is that in the macrophages, they found that, again, the insertion frequency in the stem cells was higher using the genome editing system, and that the macrophage function was actually better. There was higher levels of the Wiskott-Aldrich syndrome protein. The red is higher than the purple. And importantly, one of the functions of macrophages is to form what are called podosomes. They're areas on the membrane that allow the macrophage to do their work. And again, the function of macrophages in which the gene was edited by genome editing in red, there were more podosome positive cells than in purple, showing that at least in macrophages derived from HSPCs, they seem to have a better function than lentiviral derived gene therapy cells. And they did the same thing. They looked at the function of platelets. And again, what you can see on the top right corner is that there is more Wiskott-Aldrich syndrome protein when you use genome editing than when you use lentiviral vector delivery. The red is much higher than the purple. And when you look at the size of the platelets, the granularity of the platelets, whether the platelets are spontaneously activated or not, or whether they spontaneously die or not, we see that the platelets are bigger, that they are more granular, showing that they have better function. They're less spontaneously active, which means they won't release their contents when they don't have to, and they're less prone to die. So all of this, and in fact, the red now is equivalent to platelets derived from healthy non-Wiskott-Aldrich syndrome patients. So this is again showing that by using genome editing to knock the good copy, the wild type copy of the gene into itself, we get, again, increased safety by avoiding random integrations and increased efficacy by getting more expression in the right cells to fully rescue function. And this system has been applied across a large number of diseases now. And I list some of them here and been used by a wide variety of labs across the world. And it's exciting to me and us to see that even though we talk to each other, that the general process is highly reproducible. Now, before I go on, I just want to say, well, what about some of these other genome editing systems you might have heard about? So another way that the CRISPR system is used is to engineer the break, as I described earlier, but instead of trying to repair the break using a donor template, you just let the break repair on its own, where you can create mutations in a gene. Now, for Wiskott-Aldrich syndrome and for most of the other immunodeficiencies, breaking the gene that's already broken is not going to fix things. And there's often not going to be a workaround to break some other gene to compensate for the broken gene. So this is probably not the way that genome editing can work. Now, in the last few years, there's been two new genome editing systems called base editing and prime editing. And base editing does allow you to change single nucleotides. And in theory, this could be applied to individualized mutations throughout the gene. So if I forgot to look up how many mutations are known to cause Wiskott-Aldrich syndrome, but let's say it's like SCID, where there might be a couple hundred, you'd have to design 200 different drugs in order to treat all the patients who have Wiskott-Aldrich syndrome. Whereas the system that I described and Dr. Cavaza used, one system would correct all of the other downstream mutations. And I'll talk to you why I think this is a challenging way of going. Prime editing could also achieve individualized correction of mutations. Now, prime editing has not been shown yet to be efficient in the human stem cells, but I have faith in my colleagues that with enough work, they will figure out how to get prime editing to base editing to work in hematopoietic stem cells. But we have the homology-directed repair editing system already working very efficiently in stem cells and allowing us to develop a one-size-fits-all patients genome editing system. So how come we haven't read about these systems entering clinical trials? Well, in part, that is because it takes some time to move from the laboratory into clinical trials. But the second part is it takes significantly more money to move something from a laboratory process that you can do in a random fashion on a Thursday afternoon to in a very controlled fashion, because of course we would want the cells that were made to treat a patient to be highly to be carefully manufactured and to have careful oversight on how they're made before we gave them back. And that process costs tens of millions of dollars. And that sort of funding is really best obtained by private investors who have money to invest on behalf of their investors in terms of investing in new drugs that might make them money. And the problem is, and this is the rare of rare conference, is that investors have not considered that there is money to be made by developing transformative, potentially curative therapies for rare or ultra rare diseases, especially when the ultimate goal is to give them once in a lifetime, and then the patient would never have to receive it again. So what can we do to solve this problem? Well, I think for those of us who are in academia, we need to continue to improve the technology to make it safer, more effective, and simpler. And we need to continue to develop that pipeline such that there's pressure on biotechs and entities to finally bring these to clinic. I think all of us play a role in terms of making the process cheaper and more automated so that it would be easier and faster and cheaper to go from the lab to a patient. I think the biotech, pharma, and others really need to think about new business model for developing one-time cures for patients with rare diseases. It is possible that the current model of private companies that are have shareholders, in which the number one value is creating shareholder value in terms of money, is not the right model for developing therapies for diseases like Wiskott-Aldrich syndrome and other rare diseases. And this is of active discussion in many, many domains. No one has a solution yet, or let me put it this way. There are lots of solutions that have been discussed. No one has done the experiments testing which solution may or may not work. And finally, we need some help from our regulators. The FDA is designed to protect patient safety, and it is designed, in my opinion, to regulate drugs that might treat hundreds of thousands, if not millions of people in the United States. This highly expensive pathway serves its purpose, but for rare diseases, it is burdensome and slows things down. And I think we need to collaborate with our FDA colleagues to streamline the process to initiate an IND, which is to test a drug in the first few patients, and then a streamlined process for getting it approved so it can be given to as many patients as possible. I believe that if we defined a different regulatory pathway for diseases that are rare or ultra rare, this would be incredibly important. We also need to find a new way of making the cells that is high quality and safe, but perhaps not asking for us to pay 10 or 100-fold more than it needs to. So in summary, once we could only draw on the chalkboard in terms of genome engineering, we can now do in stem cells with high efficiency and specificity. We can really edit the DNA of a cell almost like we can edit a document on our computer. I will say that the homologous recombination-based genome editing, because it can insert large pieces of DNA exactly where you want, still has the greatest versatility compared to some of the other genome editing platforms, although technological improvements will continue. We have a need to drive innovation to lower costs and increase accessibility, not just to develop new genome editing systems. I will highlight, and again, I think this is an audience that understands this well, that we need, it would be great at some point to get rid of the chemotherapy that is needed to do a bone marrow transplant. That chemotherapy, I believe, is justified as introduced. I do pediatric bone marrow transplants, but I also recognize that this puts patients and families through too much, and it actually can limit who gets a bone marrow transplant. So developing ways to limit the amount of chemotherapy will increase the accessibility and the safety of such therapies. And I end with both an optimistic take that we have the potential to impact the lives of millions of patients. And now the question is, can we execute on that promise? So I want to thank you for your attention. I get to work with an incredibly talented group of people in my lab, high school students during the summer, college undergraduates, graduate students, PhD, postdocs, senior scientists. It's really a fantastic group of people, fantastic group of clinical collaborators in my division of pediatric stem cell transplant and regenerative medicine, work with my colleagues in our GMP facility to help us bring laboratory discoveries to patients, funding from lots of different sources, including the NIH and private foundations, and then collaborators around the world, both academically and in biotech. And one thing that I've learned in my career is that the best collaboration is when you have asymmetric expertise, where we bring one set of expertise to the table and they bring another set, and together we can create something much, much better than we could individually. So again, thank you very much. And I think I have some time for some questions.
Thank you, Dr. for providing that. Yes, we do have some time available for Q&A. But before we begin, please remember that everyone's treatment and condition are unique. The information presented during this session is not medical advice, nor is it intended to be a substitute for medical advice, diagnosis, or treatment. Always seek the advice of your physician or other qualified health provider with questions concerning a medical condition. Now, again, if anyone has a question, please submit it in the Q&A. We're actually receiving a lot of questions here, so we'll see how many we can get through.
Great.
All right. Well, our first question here is, would there be a preferred age/stage of life for this to occur?
Fantastic question. So there are two answers to that question. One is from the regulatory standpoint. They prefer that the first patients who are treated with an experimental therapies, and these would be experimental therapies, are older. They prefer adults, and then they prefer patients over the age of 12 because of the issue of consent and assent. Biologically, and from a pediatrician standpoint, I think we would want to give these therapies as early as possible in life, because as we know, these diseases can cause damage to the organs that is not always reversible. And so one would love to be able to give the therapy early in life before irreversible damage has occurred. And my vision is one day we give it so early that, in fact, the patient themselves never remembers that they had the disease. The parents will, of course, always remember, but the idea would be that the patient may never remember that they had their disease, and they would live their life knowing formally that they had a therapy, but practically not having to suffer the consequences.
All right, great. Our next question asks, how does the virus just disappear?
It's a great question. So there are some viruses, like lentiviruses, that have built into them a mechanism to insert their DNA into the genome of the cell. And that's how the lentivirus works. And then when the cell divides, the viral DNA or the gene therapy gene gets passed along and doesn't get diluted. The virus that we use does not have that protein, does not have a mechanism to insert its DNA into the cell. It just sits outside of the genomic DNA, and it doesn't have any way to replicate itself. So if you have, say, 5 copies in a cell and the cell divides, now you might have 2 copies in one cell and 3 in another. And then those cells divide, and it just gets diluted out with division. It is also diluted, gets rid of a little bit when DNA is not in a chromosome. There are proteins in the cell that degrade DNA called nucleases. And so when the viral DNA is sitting free, it is susceptible to being attacked by nucleases and destroyed. And so that's also in part why this goes away.
All right, great. Next question here is there already an animal model for gene editing in moss?
There have been multiple animal models developed to test genome editing. The most common models have been in mice. Those are the most common animal models we have used. And for example, I didn't talk about it today, but we have shown in a model of sickle cell disease that we can correct the mouse model of sickle cell disease using the process we just described. Others have shown in an animal model that you can correct the SCID-X1 version of the mouse of the human disease. It does require, however, a couple things. One is that the genomes of mice and humans are different enough that you often have to develop, you usually have to develop different guide RNAs and donor templates to do the mouse studies. So you can't use the same reagents in mouse and humans. It's not that hard to do it, but it just means that you can't perfectly extrapolate what you did in a mouse to what would happen in a human. And then the other thing is the sequence of the genes themselves are different. And so, yeah, so they have different biology. So we and others are starting to develop mouse models of these diseases because we think it could be an important way of understanding the genome editing process better. But I will say that my experience with the FDA is they don't ask us to prove that we can correct a mouse model before testing in humans. What they want to see is that we can correct human cells, and the function of those human cells work well, both in vitro in a tissue culture dish and when we put them into a mouse. But they don't ask us to necessarily correct a animal model, which is good because that is one step that does save us some time and money. Thank you.
Our next question here too. Do you intend to replace the full 502 amino acid WAS gene or only a section?
Yeah.
It was the very long WAS gene suitable for such technique, I think it means to say.
Yeah, great question. So one could replace section by section. One could design a system that replaced one exon by another. But our system actually can allow us to replace the full gene all at once. Now, we don't do a replacement. We can do that. We've done that with other genes. But what we do is we insert the full gene at the start of the natural gene. And so the way the genome now reads what we've done is it picks up the natural start and reads the gene, and then we put in a stop that says stop reading, and then downstream from that is the broken gene that's still in the genome, but we've designed it in a way that the cell never reads that part of the genome. We just ask it to read the fixed part. So we do insert the full gene. Now, our system does have a limit. We can't fix genes that are bigger than about 1,400 amino acids, but 500 amino acids is within our capacity to fix.
All right, our next question. How much longer will it be before it comes to clinic? Do you have a prediction? Will it be another 15 to 30 years?
Just kill me if it takes another 15 years. Let me just put it that way. So I will say that I hope I've vented a little bit about the frustration on how long it's taking. I do think that many of the labs that I listed as doing this work are starting to come together and think about how do we collaborate to create a system so that we can bring these to the clinic faster. I am not 100% up to date on where Dr. Cavazza is with her program, but I do know that she is doing the work to take a laboratory process and make it a GMP compatible process, is doing all the safety studies that are needed. I've seen the data, it looks really great. And so I'm hoping that in the next year or two, we'll hear that she has opened a trial testing out the process in Wiskott-Aldrich syndrome, but I don't want to put any pressure on her. So don't bug her. I know she's working hard, but I hope that we will see something like this in the next year, 2, maybe 3.
Great, thank you. All right, our next question asks, how can such a small percentage of modified cells achieve such substantial correction?
Yeah, so two questions, and I'm sorry I went through this too fast. So we are getting correction of about 60 to 70% of the cells. So most of the cells are corrected. Okay, so one, it was the, but the second thing, and one of the reasons that many of us have used these diseases as first attempts at gene editing and gene therapy, is that there's a disproportionate impact the corrected cells have over the uncorrected cells. It's a concept called selective advantage. And what we know is that in certain diseases, as few as a single corrected cell, for example, in SCIDX1, can reconstitute the immune system of a patient because the good lymphocytes outgrow in a good way the lymphocytes that have the mutant gene. In Wiskott-Aldrich syndrome, there is a mild selective advantage, but not a tremendous selective advantage. So, for example, if you have a good population of T cells, that will compensate and prevent sort of the autoreactive T cells that might develop from the Wiskott-Aldrich cells. Every disease, though, needs its own analysis. There's no one size, there's no one number that fits all. And so this is why it's really important that disease-specific and gene-specific experts work on this, because it still requires that detailed, nuanced knowledge.
All right, our next question. Are you using a specific virus vector as a delivery system?
Yes. We are using a form of adeno-associated virus called AAV. It's kind of a weird name. Don't think of it as an adenovirus. It's not adenovirus. It's a cousin. We should just call it a different name, but it has this name, adeno-associated virus. AAVs are actually viruses that do infect human beings, but they don't cause disease. About 60% of us have been infected by an AAV in our lives, but we never knew it. So they're considered a non-pathogenic virus. There's lots of different flavors of AAVs, just like there's a lot of different flavors of flu and so, and so we use a specific kind of AAV called AAV6. We have found that is the best one that works for us in the process I described. And again, I'm always happy when other people say, look, and they test other types of AAVs and they end up saying, yes, AAV6 seems to be the best.
Great. Okay, next one. How does the clinical process compare from the patient's perspective? So, for example, marrow conditioning, et cetera.
Yeah. From a patient's perspective, the difference between a genome editing and a gene therapy would be minimal at this point. The patient would still have to undergo a harvest of their stem cells. We are switching from harvesting bone marrow to doing a pheresis of stem cells from the blood, which I think is much easier on patients. But then the cells go to a lab, and of course that doesn't affect the patient's life. And so that's pretty different. And then at the end, the patient still has to go through a bone marrow transplant where they receive conditioning chemotherapy before they receive their cells. So it's a very good question in that from the patient perspective, the patient experience through the process might not be different. Now, if the patient results are better, for example, we know using lentiviral gene therapy and Wiskott-Aldrich syndrome that sometimes the platelet count still remains below normal. It's a very good level. It's enough to be safe, but it's below normal. And one of the reasons I emphasize that the gene editing approach seems to make better platelets is it's perhaps what will happen is the patient will have a better platelet count. And so the concerns and precautions one might take might be less. The second thing we will explore, and I'm a believer of you want as many things out there as possible because that allows us to figure out what's best, is that if one turns out to be better than the other, it might allow us then to say, well, we don't need so much chemotherapy, or maybe we can replace chemotherapy with something else because it works so well already. So we wanted the cells to work as good as possible because that will give us room to reduce the amount of chemotherapy.
Great. Next question here asks, would the presence of diverse revertant mosaicism in a patient affect treatment this way?
Yeah, we got a smart audience. So, revertant mosaicism is such an interesting concept, and it's from those examples that lead us to the understanding that we don't need to correct all the cells. I think if you had a diverse or even, revertant mosaicism in a small number of cells or diverse revertant mosaicism that wasn't sufficient to reverse the symptoms of the disease, that patient would still benefit from a gene therapy or genome editing approach. The mosaic, if however, the mosaicism is significant enough, then one would want to do a careful discussion about whether it was the benefit of going through a bone marrow transplant with chemotherapy is worth it. But I think that if the patient was still having significant symptoms, the presence of mosaicism would not affect the efficacy of the underlying genome editing or gene therapy.
Very wonderful. All right, next question here. How portable is such a treatment? So for how long will it be only in distant research establishments?
Yeah, that's a fantastic question as well. And that was why I raised the point that we need to make these gene therapies, and we'll just classify genome editing as a subtype of gene therapy. We need to make those processes simpler and more automated, so that they don't have to be given only in a few centers in the world. Again, I am optimistic that will happen, both because I've seen some of the developments that's going on, but also because I believe that the human species are really good engineers. When we put a group of people on a task, we can send people to the moon and we can send explorers to Mars. I mean, we're really good at building things when we know what we need to build. And I think the field is really identifying what we need to build. And I've seen some really great progress on that. However, it goes back to the chemotherapy aspect. You still would want to get the therapy in a center that knows how to take care of patients going through a bone marrow transplant, because that period after you get the high doses of chemotherapy can still be quite rough, and there's still a risk when the immune system is suppressed. So one of the other ways of opening this up is not only to streamline and make the manufacturing more automated and closing it up so it doesn't have to be done in specialized facilities, but again, is to also do this in a way that doesn't require intensive chemotherapy, and therefore at a center that knows how to take care of patients going through this process.
All right, thank you. Now I'm actually going to combine these two questions since they are similar. All right, so what can patient organizations and patient advocacy groups do to move this effort forward faster?
Yeah, great question. All of them have been great. They're fantastic questions. Thanks. I think obviously, providing financial support is helpful, but I actually think that where patient advocates and patient organizations can do some of their best work is actually participating in the process to develop a more streamlined process to get I used the word twice, but sorry, my English is a little redundant. Like I mentioned in the slide, is really participating in how do we develop safe but streamlined processes to get these therapies into rare patients, and really pushing the regulatory agencies and even politicians to think about is the risk benefit that the regulatory agencies now adopt really appropriate for these rare and devastating diseases. Perhaps we need to help them understand that the bar needs to be moved a little bit, or otherwise these will just sit on the shelf or sit in publications and not be helping patients. I think that's one really important way organizations can help. And honestly, I can tell you from personal experience, it's great to get a pat on the back when you go to a meeting, and science is hard, translational science is hard, having a disease is hard, and I don't want to compare what we go through to the patients and families, but we all know that sometimes a pat on the back can go a long way.
All right, so we do have more questions coming in. This is a great group. I'm not even sure we're going to be able to get through all these questions here.
Okay.
But next one here is, will this be available to patients with a milder form of loss?
Yeah.
So going back to your previous point.
Yeah. So when these new therapies get developed, there's I would say, a negotiation between the investigators and the regulatory agencies about how severe the severity of the patients before they're eligible to get the therapy. And that makes sense to some extent. You don't want patients who are so sick that they might get even sicker from going through the process. However, you don't want patients who have such mild disease that they're taking undue risk by going through the chemotherapy. So there's a sweet spot at the beginning. However, if the process is shown to be safe and effective in patients with severe disease, then there's the argument that even if somebody has a milder disease, they still have the disease, and the risk-benefit equation shifts. And so then you can make the argument that this is the appropriate therapy for patients with milder disease.
You also assume that you use the selective advantage as in lentivirus therapy.
We do assume that. I guess I wouldn't say assume is we take advantage of it. And we hope that right now, both gene editing and lentivirus therapy, there's been no demonstration that one gives more or less selective advantage than the other. We take advantage of the biologic selective advantage. I will say in some of the other diseases we study, there is no selective advantage. And in those cases, we have to determine, again, maybe the frequency of correction needs to be higher because there isn't a selective advantage, or we have to figure out ways of increasing the levels of correction. I didn't include it in today because I wanted to leave plenty of time for questions, and I'm glad I did. But in addition to new genome editing technologies being developed, we and others are developing ways of increasing the efficacy of the genome editing, so that instead of only having, say, 50% of the cells corrected, we can get to 80% or 90%, and maybe one day even 100%. So those are ongoing areas of investigation. So thanks.
Great. Now, again, we've got about 5 minutes left here. So let's see if we can just get through a few more.
Yeah.
All right. Meeting FDA safety requirements designed with different, more conventional therapeutics in mind, what new/different protections are needed for individuals and families paying for novel, or unproven rare therapies?
Boy, that's a there's a couple. I think there's a couple questions in that question. I think in terms from the FDA standpoint, is coming to an agreement that if you have, I'll give an example. If we have a molecule that we pay $10 for, and I'm going to use intentionally wrong numbers because nothing costs $10 that we use. And it works just as well as a molecule that costs $1,000. Currently, the FDA requires us to use the $1,000 molecule, even though it's identical to the $10 molecule. So that's one way the FDA could help us. I think if I understand the second part of the question was, if you are a patient on a clinical trial, then of course you should not be paying for any part of the care because you have taken, you should not be paying part for any of your clinical care. Once, and I'm going to be an optimist because we have to be, once these therapies are approved and then they are charged, then there is a charge for them. Like any other medicine, how to pay for what are turning out to be very expensive medicines is an important issue. For children in the United States, and again, I feel like I might put my foot in my mouth here, but most children in the United States with a disease like Wiskott-Aldrich syndrome can get Medicaid coverage, and then they would pay for it. However, not all do, and of course that there is a huge gap for adults. And so how these therapies are going to be paid for and can our healthcare system afford them is important. Now, again, I'm a pediatric stem cell transplant doctor, not formally a pediatric immunologist, but I count them among my friends. And I personally believe that even if these therapies are or the charge is $2 to $3 million, which is the current amounts people for similar therapies are charging, I believe that's worth it to save a child from the suffering of their disease for their lifetime, to reduce the stress and burden on the families and patients around this child. I think that price is worth it. But there is a sticker shock when you say that medicine is $3 million. It makes us all sit up and go, really? On the other hand, I think when you do the analysis, it's worth it.
And I think we have time maybe just for one more question. So, all right, what is your estimation regarding the stability of the fixed cells in the long run?
Yeah. So from allogeneic bone marrow transplant, so when we do a bone marrow transplant from somebody else, we have seen that the stem cells have lifelong durability, that they can last for the rest of one's life. What we don't know yet is if we genetically manipulate the patient's own cells outside the body and then give them back, will they have lifelong durability? However, we know from some other forms of immunodeficiency that we have cells that have been given back to patients now, I believe we're up to 25 years, and they're still persisting. And so every year that goes by, we'll have a better answer to the question, are these truly going to be cells that have lifetime durability? The expectation that they will, we do everything we can that they will, and the biology says that they will, but the proof will be in the patients.
Great, thank you. All right, well, I think we're going to have to come to a close here. Thank you again so much, Dr. Porteous. It's really been an honor to have you join us today. Everyone, this session is one of 39 unique learning opportunities provided through the conference. So we hope you continue tuning in today as we explore other critical topics for the PI community. Have a great day, everyone.
Thank you. And thank you for all the great questions.
Bye.